Hello everyone, myself Vincy Varghese, Department of Pharmaceutics, Nirmala College of Pharmacy, Noida. Today we will discuss about the gram staining of bacteria. So this method was first developed by Danish physician Dr. Heinz Christian Gram in 1984. And this method is mainly used for the differentiation and identification of bacteria. We can differentiate bacteria based on this gram staining procedure into two groups, gram positive and gram negative. So next, coming to the steps involved in gram staining method. There are mainly 6 steps involved in gram staining method. First one is the smear preparation, or that is the slide preparation. Then second one, heat fixing of the bacterial smear. Third step, primary staining with crystal violet. Fourth one, use of a mordant, that is iodine or Leupold's iodine solution. Fifth step is treatment with a decolorizing agent, that is acetone alcohol mixture. And finally, using a counter stain, that is safranin. These are the steps involved in gram staining method. Next is the reagents or the stains using for this gram staining method. First one, primary stain, that is crystal violet. Crystal violet is used as the first step, and it will stain all the cells deep violet in color. It gives a deep violet color to the cells. That is the primary stain, that is of crystal violet. Second one is the mordant, grams iodine or Leupold's iodine solution. This mordant is not a stain, and it forms an insoluble compound with primary stain, that is crystal violet, and it will fix the color of the primary stain to the bacterial cell wall. That is the main purpose of using a mordant. Next is the decolorizer, that is alcohol or acetone alcohol mixture. This decolorizer is mainly used to remove the primary stain and the mordant complex, that is crystal violet mordant complex, to remove the complex from mainly from gram negative bacteria. That is the purpose of using decolorizer, to remove crystal violet mordant complex from gram negative bacteria. And this decolorizer, it is composed of an organic solvent like ethanol, acetone, or combination of this ethanol acetone. That is the component of decolorizing. Then finally, counter stain. We are using safranic as the counter stain for gram staining method. Counter stain is mainly applied to stain those cells that have lost their primary stain, mainly in gram negative bacteria, as a result of decolorization. That is the purpose of using counter stain safranic. So these are the reagents using in gram staining method. Next, we will discuss about the principle involved in gram staining method. So, in gram negative bacteria, the gram negative bacteria, they have their cell wall is very thin, and they are multi-layered, containing high lipid contents. So, when we are adding the decolorizer, that is alcohol, the lipid contents are readily dissolved, and that results in about pore formation in the bacterial cell wall in case of gram negative bacteria. And this from this pore formation, it will facilitate the leakage of primary stain mordant complex, that is the crystal violet iodine complex. This complex will leak out from this pore, through this pore. And this results in discoloration of gram negative bacteria. And this takes safranin and appears red in color. In case of gram negative bacteria, the bacterial cell will appear red color. That means it will take the color of counter stain we are using, that is safranin, due to the result of pore formation in the cell wall, and through that pore the crystal violet iodine complex came out from the cell wall, and it will take the color of the safranin, that is the counter stain we are using. So in case of gram negative bacteria, the bacterial cell wall will appear as red in color. And in case of gram positive bacteria, the cell walls are thick compared to gram negative bacteria. So it is composed of mainly proteins and cross-linked mucopeptides. So upon application of the decolorizing agent, that is the alcohol, dehydration is caused. And this results in the closure of the pores in the cell wall, bacterial cell wall. And thereby the crystal violet iodine, that is the primary stain, mordant complex will retain in the bacterial cell wall, and it appears as the color of the primary stain, that is crystal violet. So in case of gram-positive bacteria, the bacterial cell will appear as blue or purple in color. That is it will retain the color of primary stain. So that is the principle involved in gram staining method. In gram-negative bacteria, the bacterial cell will appear as red color. That means gram-negative bacteria, the cell will take up the color of the counter stain we are using. That is the red color. And in case of gram-positive bacteria, the bacterial cell wall will appear as a blue or purple color. That means it retain the color of primary stain. So this is the principle of staining technique. That is in gram positive, and this is the gram negative bacteria. First is we are using primary stain crystal violet. Then next we are adding mordant, that is iodine solution, then alcohol, decolorizing agent, alcohol or acetone, and counter stain safranin. So in gram positive, first we are taking the bacterial smear, that is the bacterial slide, heat fixation. Then we are adding primary stain crystal violet. So in gram positive and gram negative, it will take the stain of primary stain, that is crystal violet. Then we are adding iodine treatment. Then next, decolorization, that is adding alcohol acetone mixture. So in gram positive, it will retain the violet color, and in gram negative, in gram negative bacteria, the crystal violet iodine complex, that is leaked out from the gram-negative bacteria. And finally, we are adding the counter stain, that is safranin. So in gram-negative bacteria, it will take up the color of this safranin. That means the cell will appear as red in color. And in gram-positive bacteria, the pores are closed and it will retain the color of primary stain, that is blue or violet color. Then this is a brief explanation of the procedure for gram staining of bacteria. First, we are adding gram positive. First, we are adding crystal violet, that is the primary stain. Then we are adding mordant, iodine solution. Then alcohol, that is decolorization. Then finally, we are adding the counter stain, that is safranin. So in gram positive bacteria, the bacterial cell will appear as color of the primary stain, that is blue or violet color. And in gram negative bacteria, the cell will appear as the color of the safranin, that is the counter stain we are adding, that is a red color. And again, a brief explanation of the procedure for gram positive bacteria. First is the bacterial smear preparation. Then we are adding crystal violet, that is the primary stain. So violet color appears, and then decolorization, that is iodine or grams iodine solution. And this iodine acts as a mordant. Then we are adding what with alcohol, violet color persistent. Then we are adding finally the counter stain, that is safranin. And in case gram positive bacteria, the violet color will remain. And in gram negative bacteria, first bacterial smear preparation, then crystal violet, then mordant, then decolorization with alcohol, and finally safranin. And it will take up the color of safranin, that is a pink or red color will appear in case of gram negative bacteria. And this is the observation. For gram negative bacteria, it appears as a pink color, and for gram positive bacteria, it appears as color of the primary stain, that is blue or violet color. So that is about the gram staining methods. Thank you.